Hi, everybody. Danny, can you hear me?
I can.
Hi, welcome. Welcome to everybody. I want to just see, let's just give one more moment. Just one sec here. And I guess you'll be able to just share your screen, right, Danny?
Yes.
Okay, just one sec. All right, I think you can go ahead and share your screen, and I will go ahead and just give a very brief introduction, if that's okay. So I want to welcome everybody, tell you a bit about, we're so happy to have Danny here. She received her undergraduate degree from University of Washington and her PhD from Yale, and did postdoc work in genetics at UPenn. She joined Monell Chemical Senses Center faculty in 2000. She's now a member and the associate director. She's also a founding member of the Global Consortium of Chemosensory Research and is the current president of the Association of Chemoreception Sciences. And I've interacted with Danny quite a bit myself because we share an interest in the genetics of taste perception. So I'm really looking forward to your talk, Danny.
Yes, well, thank you so much, Sarah. And it's really wonderful to have the opportunity to talk to your group. For those of you who are geographically based in Penn or nearby, we are geographical neighbors. If you've ever gone down Market Street and seen the Golden Nose at about 35th and Market, there it is in all of its glory. Our tagline is, when it rains, it drips. So it's a good landmark for you, and we welcome visitors, and we'd love to have you visit if you're at all interested in taste and smell or any research along those lines. And I just wanted to give a shout out to the members of my lab, and particularly I wanted to give a shout out to Katie Bell and to Chris Jones, who are both, Chris is now Sarah Tishkoff's lab and was a very beloved member of the Reed Lab for a while, and Katie Bell, I want to just give a shout out to her. She really did a tremendous amount of work for the research I'm going to talk about, and also to Ha Nguyen, who I see is tuned in today. So I'm going to be talking about this idea of worldwide taste research. And so I'm going to provide you with some background about taste. I'm going to tell you why we think it's important, and particularly we're going to be focusing along on the taste of medications and the potential implications of that for worldwide health. And so that's sort of the direction that we're going. So just to give you a little primer on the sense of taste. So taste does indeed start on our tongue. And for those of you who are looking directly at your screen, you can see a beautiful pink tongue that's dyed blue with the fungiform papillae. Those are fungiform taste papillae, those little pink dots. And so the papillae are visible to the naked eye, and they house the taste buds, which you'll see here on the left. That's an orange-shaped structure that has different taste receptor cells in it. And we actually use the phrase taste bud when we're talking about this. So taste buds are not visible to the naked eye, but they're the unit of taste perception. And you'll notice that taste cells have a kind of interesting morphology. They are surrounded by the epithelium, but they have little cilia that stick up at the top. That's the taste pore. And that's where things in the mouth interact with the receptors. So right at that point. So we're just going to take a little tour of the tongue. So we're headed back here. There are three types of taste papilla. One is the circumvallate. Those are those big bumps that you see right there. So those are in the back. Those are very sensitive to bitter, kind of a gatekeeper. You're looking here at the sides. This person I chose because she has nice pigmented foliate. Those are folia, leaves on a book on the side of the tongue. And then underneath the tongue is an interesting area because it has no taste receptors. And so often people will put bitter medicines under the tongue to dissolve because you get sort of less of that bitter taste. So side of the tongue, foliate. If you've ever put anything sour in your mouth, you kind of, sometimes that engages the foliate. You get that sort of soury, saliva releasing response. And here I'm showing you the soft palate to remind me to say to you that there are taste receptors on the soft palate of the roof of the mouth as well. So from based on what I've just said so far, it'll be, it should potentially be clear that there are a lot of chemicals in our food and medicines, and we don't like to think of our food as chemicals, but in fact, of course, that's exactly what food is. And these foods and medicines taste bitter, and there are specialized receptors on the tongue that are used to detect these bitter compounds. So just a little bit more about these bitter receptors, because they'll be actors in what will come in the moment. So there's a whole suite of bitter taste receptors. So I'm going to talk about them using different names. And so I just want to say what those names are, so if you're not confused. I'll talk about bitter receptors. I'll talk about TAS2Rs. So that's TAS is for taste. The 2 is because it's the second family, with the first family being much smaller sweet receptor family. R, of course, for the receptor. And sometimes we just call them sort of affectionately T2Rs. So that's, they're G protein coupled receptors. So they weave in and out of the cell membrane of the taste receptor cells, some parts touching the outside of the cell and contact with the food and the medicines, and some parts signaling inside the cell. And then here I've also shown you a phylogeny. So these taste receptors, they have a very pesky history in terms of nomenclature. And you'll notice that there are roughly 25 of these taste receptors. And I say roughly because actually some of the taste receptors are missing in some people. So roughly everyone has 25 taste receptors. And you might logically wonder why, if there's 25 of them, why they're numbered up to 60, and some numbers are missing, and why is this so sort of disorganized? And the reason for that is when the taste receptors were first discovered, it was the early days of people using bioinformatic methods and looking at the human genome for the first time. Everybody got very overexcited and kept finding these taste receptors and naming them and not consulting each other. And at the end of the day, the Human Gene Nomenclature Committee, I think, just didn't have the heart to really straighten everything out, because there was such permeation of these particular names in the literature. So I explain this to you because it can be very vexing for trainees trying to understand the numbering system. And you'll also notice in this phylogeny that I'm showing you, there are some solid lines and some dotted lines. And the solid lines are all receptors that are on human chromosome 12. And the big dashes are on human chromosome 7. And you can see one lonely little receptor here, which is TAS2R1, the first receptor, and that is the little dotted line. So the point is that these occur in clusters on the human chromosomes, and they're predominantly on chromosome 7 and chromosome 12. Now, it's very interesting because the taste receptors are really subject to a lot of pruning by evolutionary forces that guide how and what animals taste. And we see this reflected in variations on their taste receptors. So if you look on the screen up to your left, you'll see a picture of me sitting quite comfortably with a panda. And this panda has, unbeknownst to people in the beginning when people were studying panda, because of course pandas are famous for eating bamboo, and they have a whole suite of bitter receptors to guide them to the sweet and the good bamboo and to keep them away from plant foods that are toxins. But it turns out pandas also have the sweet receptor, and if you're a good detective, you'll see that this panda, although it looks like it's enjoying chatting with me, is actually being bribed to interact with me by holding some sugar cane in its hand. So that little stick is actually sugar cane. Now, when we think about sweet receptors, one of the things that my laboratory discovered about 10 or 15 years ago is that big cats like lions and tigers, but also small domestic cats like the cats in your home, actually have broken sweet receptor, so that the sweet receptor is actually non-functional. It's a pseudogene in the cat. And this makes sense evolutionarily because cats are obligate carnivores. And so they have no need to sense out and ingest sweet, and actually sugar can make cats quite sick. And this was an interesting just experience for me personally, because this was the first time that I had ever gotten hate mail from the general public when we first released this result, because people were so outraged, writing and telling me their cats eat ice cream and their cat eats pie and their cat eats cake. And of course, sort of overlooking the fact that cats love fat and are certainly very attuned to that. So that was an introduction for me as a young scientist, one of my first interactions with the general public. And the other thing I'll just mention is that some animals, particularly sea lions and dolphins, when we looked in their genomes, all of their taste receptors are broken. They don't function. They're all pseudogenes. And that I guess makes sense if you think about actually how they eat, which is by they swallow their food whole. So they're not sort of chewing and sampling, but rather they're just recognizing their food visually and sucking it in. So it's logical, I guess, if you think it from that perspective, that they wouldn't necessarily need their receptors. And I tell you this, because it's a bit interesting. And I also tell you this was the idea that evolutionary forces probably shaped the human bitter receptors in much the same way. Now, the most important thing I could tell you about human bitter perception, bitterness of foods, bitterness of medicines, the most important thing I can tell you is that people are really different in how they perceive the bitterness of many foods and medicines. And this is an experiment that we did a few years back where we asked people to chew raw vegetables and spit them out and rate their bitterness. And what you can see here on the y-axis is just the people's rating of the bitterness of various vegetables. So big numbers mean something is rated as more bitter. But what you see along the x-axis is this is the variation, the standard deviation, the degree to which people are different. And you can see something here, which has probably informed a lot of dinner party food selections, which is carrots are not very bitter. People don't rate them as bitter, and everybody pretty much agrees that they're not bitter. But when we get into other territory, culinarily speaking, so collards and watercress and Brussels sprouts and endive and bitter melon and so forth, you can see that not only are these things on average fairly bitter, but there's a great deal of person-to-person variation in this bitterness. So there's a lot of difference of viewpoint about it, which makes one of the things that can be hard to find something everybody likes. And I sometimes give up in despair trying to find things that everybody enjoys at my house. And of course, this is logical when you think about it, if everyone is experiencing their sensory world in a slightly different way.
Danny, can I ask a question?
You certainly can.
So in that last slide, I'm assuming there's individual variation, but have you looked to see if there's population variation for how people perceive these different foods? I'm just wondering if it explains different diets and different populations or regions of the world or something like that.
So we'll be taking a deep dive into population differences in bitter perception, but not with foods. So I think this would be a fantastic experiment to do with foods, and I'm totally on board if you want to think about that, because it would be really interesting to see population differences, not just individual differences. So given that I've sort of paved the way for you by telling you that the bitter receptors, describing bitter receptors, and describing the relationship of bitter receptors and the sense of taste, it'll come as no surprise to you at this point that a lot of this individual variation in bitter perception we think is due to variation in these taste receptors. So we'll just explore that point right now. So I'm going to use as my example quinine. Now, of course, Sarah is very well known for her work with PTC, which is the sort of the textbook standard of human variation in PTC. But PTC, phenolthiocarbamide, in that classic example of bitter perception, is not necessarily an exception, but perhaps more like a rule. So here I've given you some data on quinine. So we'll remember that quinine used to be a first drug in the treatment of malaria, long since outpaced by other and better drugs. But quinine is actually perceived as not very bitter by people with certain genotypes of one of the bitter receptors. So remember, the bitter receptors are these TAS2Rs. this one happens to be 19. And people with this GG genotype don't find quinine very bitter at all. Now, this VAS and LMS are just different ways that we ask people, different scales that we use to have people rate bitterness. And I just wanted to make the point that this is highly reproducible. So we published a GWAS study, but this is a replication from another group. Now, we actually made this discovery in a very interesting way. So one of my laboratory workers is a woman from Japan, and she came into my office one day and she was very tearful because she thought she'd made a very serious error in the lab, and she thought she had just failed to include the quinine in some solutions that we'd been using, and she was very distraught. And she was telling me that this quinine tasted like water. And of course, I tasted it and I spit it right back out because to me it was excoriatingly bitter. And it turned out really when the dust settled on this project that she had had this GG genotype and she just couldn't taste the quinine. So she'd made the solutions absolutely correctly, but it was really a hint. It really foreshadowed what we later learned, that this is a genetic trait. So when we're thinking about diversity in human taste response, we were very much influenced by Sarah's paper on the missing diversity in human genetic studies. So we really reflected on what we had learned from this paper, and we thought about our field, and we were curious as to see how we were doing with looking at diverse populations in the field of taste, because Sarah's already expressed interest in knowing about, we're very sensitized to all the beautiful world cultures when it comes to food. And so we're sort of used to thinking about people's tastes being different worldwide. So how well are we doing with actually studying this? And as you might imagine, we're doing quite miserably. So this is work that was done by a postdoc in my lab, Han Nguyen. And she did something very clever, which is she just went to one of the premier taste and smell journals, and she scraped all of the data for certain years. And she just asked the question, where are the participants being studied in these taste and smell studies? And you can see here that there's an overwhelming preponderance of participants that come from America and a little bit from Europe, but areas of Africa and Asia are sadly neglected. And so there's really missing this diversity, deluxe missing diversity in the areas of taste and smell. Now, when we think about taste and smell, it's very topical and very much on our mind that taste and smell loss is a cardinal feature of COVID-19. And so one of the implications of that for me was that, taste and smell scientists were very earnest and hardworking, and I like to think we're dedicated to our job, but we were really catapulted into the national spotlight because people were so curious, distraught, and about these very mysterious symptoms with COVID-19 and this loss of taste and smell. And actually, this is a very sort of a beautiful thing happened right at the very, very beginning. I remember it was in the middle of March, actually, when we were just starting to get reports, March of 2020. Taste and smell scientists were hearing these strange reports about the loss of taste and smell. And people came together worldwide through the use of Slack, which is one of these wonderful tools for worldwide collaboration, and formed a global consortium for chemosensory research. Now that's a mouthful, but it's exactly what it sounds like. You can see the scientists that participate in this. It's a little bit more representative than the participation of the research subjects I just showed you. But these are scientists who came together to try to study taste and smell loss. And this is the development of this study of this in the sort of frantic days of the early pandemic, and studying this very quickly and very, and trying to understand very fast, was reported in a New York Times article here, which I showed you here. And I just wanted to say a few things. One thing I wanted to say about this is this article features me very prominently. And it was a bit of a shock to me because I learned that this is the first time I'd ever been sort of in the New York Times. And it was quite exciting and a little shocking, but I learned two things, which is when this type of publicity happens, you get invited to serve on a lot of advisory committees all of a sudden. And the other thing I learned is that old boyfriends get in touch and want to talk. So it was definitely like a very unusual life experience. So I should say that one mission of the Global Consortium for Chemosensory Research, or the GCCR, was we really wanted to try to understand this taste and smell loss in people worldwide. We really were very sincere in our effort about this. And so I just let me back up here and just say, I was talking about taste and smell loss. And so one of the controversies is that people often confuse smell with taste, particularly people without necessarily any training in this. So a lot of times people will say, oh, I can't taste my food, when actually what's happened is they've lost their sense of smell. And so they're not getting the aroma, but their sour, sweet, salty, bitter coming from the tongue is just fine. So there was a bit of a controversy because people were reporting taste loss with COVID, and sensory scientists were skeptical about this because it's very, very rare to lose your sense of taste, very rare. Losing your sense of smell is very common, but losing your sense of taste is very rare. And it turns out that with all of the studies that were done in the early days, especially studies where people were directly measured for their sense of taste on the tongue, so that's the directly measured studies. Here, we can see that about 44% of people did report losing their sense of taste. Now, these studies are from the earlier days when it was alpha and beta and even delta. So with Omicron, there's much less taste and smell loss and much less taste loss, but so many more people are affected. Actually, the numbers are roughly equivalent. So the main point here is that we were able to show that it's not just smell loss, but it's true taste loss with COVID. Now, as I said, the Global Consortium was really earnest about trying to study this problem globally. And so we translated our survey into 32 languages, and we were surveying people who had had COVID, and we were wanting to know about their loss of taste and smell. And so one of the things we learned is no matter, we tried really hard, I guess what I want to say is we tried really hard to get global representation, and it was a big failure. And I say that because you can see here the people, we did a survey of 15,000 people, but most of the people were from the US and from Europe, despite the fact that we had translated this into 32 languages. And this really was and if you look at the bar graph on the right, you can see that the GCCR participations are mostly that tan color, mostly people from America and Europe. And you can see the people that actually live in the world or people that actually had COVID is a much different representation. Trying to make the point here that this has really foreshadowed some of the difficulties that we encountered as relative novices trying to do global research. So why do we want to do global taste research, and why does taste matter? So there's many reasons for this. Sarah just touched on one about human food selection. That's a really important thing for people's health and well-being, absolutely. But another place where taste is very important and is often really overlooked is the taste of medicines. And one of the things that is something people don't necessarily talk about is how the bad taste of medicines influences whether medicines are taken as they're prescribed. So here is a recent article in the New York Times, and I'll talk to you a little bit about that in a moment. But I'd just like to recount some personal experience I had. So, and those of you who have had our parents or have given medicine to small children, I had a great deal of difficulty getting my children to take medicines like antibiotics that were bitter tasting and nasty. And it's not something I felt necessarily, I felt kind of ashamed that I had so much difficulty, and was sort of reluctant to talk to my pediatrician about it because it just seems like it should be easy. And of course, it's really not, because kids, especially liquid formulations, they fight, they don't want to take it. Some of them choke, some of them vomit. It can be very difficult. And it's particularly difficult for children with HIV because they take, many children have to take liquid formulations, and that those taste very bad indeed. So one of the things that this just struck me reading the New York Times last week, where we do see attempts to make these medicines taste better. And this is just an example of trying to flavor a medicine to make it taste better and not really being very culturally aware. So this formulation of a new retroviral medicine was formulated with strawberry flavored syrup. And of course, I was just wondering, and many of you will know better than I and have been far more experienced about this, but I just wondered, are strawberries a thing in Kenya? And it turns out that they're not. They're one of the bottom in consumers and producers of strawberries. And so I just wonder, you know, we're not necessarily, this is an example, I wonder, of formulating medicines to European palates and perhaps not the palates of the people most in need. So I just want to remind you about these individual differences in bitterness, and particularly of medicines. I gave you this example of quinine. And what I didn't tell you is that these, the rates of this, remember this G genotype, this is the genotype that's essentially blind to the taste of quinine, has different representations in different parts of the world. So you can see here, people in some parts of the world are going to be on average more sensitive to quinine than people in other parts of the world. And you can just get a sense of this by looking at this worldwide map. And it really gets at the question of, you know, there can be logical and genetic reasons where people's tastes might differ by their ancestry, their geographic ancestry. So we have undertaken a study to study this, and we do have this international case study that's ongoing. And we are studying these medications here, moxifloxacin, imodiaquine, praziquantel, and tenofovir. Tenofovir, I'll just hold up for you because that's an anti-retroviral. It's used treatment in HIV. It's one of the active ingredients in a lot of the liquid formulations. And so we asked the question of, does the taste of these medications differ in people worldwide? So this is just a little bit more information about these medications and what they're intended to treat. So, and again, we'll be focusing mostly on tenofovir, or we call it TAF. So when you think about it, we started this project in 2020, and I've always greatly admired Sarah and her research program and traveling to Africa. So it sounds very romantic and a lot of hard work, I know for sure. But this was just not in the cards for us. To travel worldwide and collect these data was just not going to be possible, although that is a passionate dream that I have. Instead, what we decided to do was to essentially capitalize on the changes we saw in the research climate based on COVID, which is we made use of Zoom. So we recruited people that are recently immigrated from different countries worldwide, so that they had been living in those countries and had moved, or their recent parents had recently moved from those countries. And we sent them these taste stimuli to their home, and then we supervised them, watching them remotely by Zoom. And so this was a way of sampling the worldwide, and really it was a way that we could do this comfortably and our participants could be comfortable. And one of the things we also learned is that we really needed a special effort to recruit people from sub-Saharan Africa. So we recruited immigrants from Southeast Asia and Europe, and things went very speedily, but we had a great deal of difficulty recruiting people from sub-Saharan Africa. And I also just want to shout out again to Sarah for her help and advice and moral support in this. And it was a really humbling and it was a learning and teaching moment for me. And one of the things we learned is why all of these wonderful social media things worked great. We have a beautiful flyer, Twitter, we have a website, we advertised on Craigslist and Instagram and Facebook and Reddit and all those things. And those worked great for our other ancestry groups. But for our sub-Saharan African group, the only thing that was very effective was what I'll call professional networking and having previous participants recruit and help us. And so it was a very deep learning for us. And we're really still in an infancy of learning how to do this type of work, but we realized that we needed to change our recruitment methods to be effective here. So Our recruitment is now complete. So that was a big red letter day in the lab. So that happened in December of 2022. So that was a much celebration. And we're in the process of analyzing our data. And so here's what I can tell you so far. So remember that I told you we were giving these 5 medications: moxifloxacin, praziquantel, TAF, imododiaquine, and PROP. And PROP is akin to PTC, and it's kind of our gold standard for individual differences. And what you'll notice here is that these groups are very crudely represented just as European, Asian, and African. And we'll be doing much more nuanced analysis as the genotyping data is available to you. And I'll say a bit more about that in the moments to come. But the real take-home message to me is we entered this project thinking that we would see differences on average between people of different ancestries, and that is not what we see. Instead, it's really a story. There's some differences, ancestry-based differences here. You can see the little stars here denoting the group differences. But the real message is how deeply and individualized the sense of taste is. So if you just concentrate up on TAF on this upper plot here, you can see that there are some people that find it to be excoriatingly bitter. They're at the very top of this scale, whereas other people are just absolutely insensitive to it. And so you can imagine, as you're trying to make medicines that kids and other people are going to take, some people are going to be fine, right? They're just going to have no problem at all. And other people are going to be just reviling this medicine and choking and gagging, and it's just going to taste awful for them. So
Danny, can I ask another question?
Yes, of course.
The one on the lower left, which I can't pronounce,
imododiaquine, yeah.
That does seem like it shows some difference. Remind me, what is that one for?
Imododiaquine is malaria. And
okay.
Yes, I don't want to say that there are no ancestry-based differences in the aggregate. You're quite correct, there are. And those were probably going to be more revealed to us as we do a finer grained analysis of the ancestry, for sure. I don't want to mislead you. But I also want to make the point that within every population, people are very different. So I just wanted to say, give you a progress report on how we're handling trying to evaluate ancestry. So we ask ancestry by self-report, and I will explain that to you in the next few slides. But we also are doing a DNA-based analysis of ancestry, and we're using 2 methods, ancestry informative markers, and we're also doing principal component analysis. So for the ancestry informative markers, we have about 2,500 markers that really are good at discerning different ancestry groups, and I'll show you a little vignette of that in a moment. And we're doing principal components analysis, where we take advantage of all the marker data that we have. And I know this is a genetically quite sophisticated group in the aggregate, but just in case this is not familiar material for you, when we talk about markers, we're really talking about places where there's specific locations on a chromosome where people differ. So when we were doing self-reported ancestry, we approached this by asking people where their grandparents were born. And that turned out to be a naive approach, an interesting approach and fascinating, but somewhat naive, because in my mind is that people were immigrating from particular countries, and I just really didn't understand the complexity of every human story. And so we do have these self-reported data, and you can roughly say, for instance, this person here is an immigrant from Africa, but of course it misses the nuance of the different geographic locations. So self-report is what it is, and this is a method that we use to do this type of ancestry. But we also know that we're going to be relying on the DNA-based analysis to make a finer grained distinction of groups. And so we're using these ancestry informative markers. And this is just a little vignette showing you that if for each alphabet character is a person. And so some markers are very much uniform in people of African ancestry or Asian ancestry, but they're very different, for instance, in Europeans. And then with the principal components method, it's not so much in any individual markers particularly informative, but in the aggregate, people that are more similar in genotype are more similar from their geographic ancestry. And so when we do the ancestry informative markers on a subset of the people, so this is an interim data analysis, we can see that there's some, it sort of depends on how you look at things. There's people are roughly coming from where this is their self-reported category here. You can see that there's also a lot of other things going on. So we have people that are reporting they're from Europe, but they have a lot of African ancestry by these markers and so forth. And so we're really learning how to best analyze these data now. And we're also taking a look at our principal components analysis. And here what you're seeing is a principal components analysis of very early data. And seeing that people and where the labels are where people say their ancestors were from, and the principal components result is based on their DNA reports. And it sort of depends on whether you think the glass is half empty or half full. We do see some aggregation of between Asia, Southeast Asia, Europe, and Africa, but then there's a lot of interesting exceptions. And it really reminds me that everybody does have a story, and even though we like to think categorically, the world is extremely quantitative. So just to drill down a little bit on the genetics. So I told you that the person-to-person differences in bitter perception of medicines are likely due in part to genetic differences. And so let's just have a little bit of, if you'll allow, just a little taste of this type of analysis to see if there's any genotypes that can predict these phenotypes. So remember I told you that TAF is a medicine that's used to treat HIV. It's used as a liquid formulation. Tastes very nasty. So this tenofovir actually stimulates 4 of the human bitter receptors, 1, 8, 14, and 39. And so one of the things we might expect is that there might be genetic variation within one of these receptors that explains this differences in taste. And preliminary evidence suggests this is the case. So these are people that are genotyped for TAS2R8. Remember, I just told you that's one of the receptors that responds to TAF. This is the overall bitterness ratings here. And so we can see that there are some genotypes, which are those would be the AA genotypes to your far left in the light gray. People who are reporting that they really can't taste this TAF, which is, remarkable to me because it really is extremely bitter. And so one of the hopes here is that we're going to be able to understand exactly why people perceive these medications differently. And as we formulate and add bitter blockers to try to make the medicines more palatable, we'll be able to have a better understanding of who needs these types of blockers and who doesn't. So what is the bittersweet truth that I alluded to in my title? So the bittersweet truth is that person-to-person and group-to-group differences exist. And I guess the way I'm thinking about this currently is that there is so much individual differences, and the idea of needing to customize or at least offer choice in medication formulations is very important. So for people that don't find the medication very bitter, they don't need all of the blockers and the types of tricks that we can and are developing to do, whereas other people really are going to need the full arsenal and all that entails. So this also left me with the knowledge that collecting data that truly generalizes to all the people of the world is a daunting task, and we've certainly been on a steep learning curve of how to do this. I don't think we've done it particularly well, but we've done it with enthusiasm and with humility and as a learners, honestly. I really have a big dream to study taste worldwide. I would love to be able to actually go to different countries and to be able to do this hands-on as we would in a sensory lab. I think that would be fabulous. Person-to-person differences in all the things we've studied is the rule rather than the exception. So whenever you're puzzled at a dinner party or you're trying to get somebody to take their medicine, please remember that. Not everybody perceives the world in the same way. And again, as Sarah pointed out, that doesn't mean that there are not ancestry-based differences. We see this all the time in the food and beverage companies, because they often formulate to country. So if those of you who visited Mexico or the Europe will understand that the sweetener formulations of sodas are very, very different country to country. If you've been to Mexico, it's very sweet. And if you've been in Europe, you know that the sweetener blends are very different than the sweetener blends that we have here, very different. And I think the final part of the bittersweet truth is that learning to recruit people from sub-Saharan Africa, immigrants, really required a lot of trust on their part and our part. And we did a lot of listening, and we really have brought humility to this process as essential recruitment skills. It was much harder than we thought, and it really required us to really grow as scientists and individuals. So with all of that said, I'm very delighted to have been able to talk to you today, and I'd be very happy to answer your questions.
Thanks so much, Danny. I'm sure everybody's clapping. Yes, I want to open this up then for discussion. I have some questions, but let's see if others do as well. And feel free to just unmute yourself.
I just also want to say a shout out to Maya. I see Maya here. Hi, Maya. Thank you for coming.
And at the end, I will give a comment. I don't have a question, but I'm
okay. You can do a comment. Go ahead.
Yeah, I'm basically, as Danny knows, and both Sarah and Danny and some other people on the call know, I love to host dinner parties. But after your talk, I'm really, really scared about that. It is just shocking what the differences are within ethnic group. It is absolutely surprising. And I wonder whether we should make an effort to actually learn more about some unique aspects of our taste, so that, you know, when people ask us about food, not preference, how do you call it, food restrictions, that we say something, you know,
Are you saying, Maya, that we should ask for genotype results for their, you come to a dinner party and I'm going to genotype you?
That would be the easiest. Yeah. That would be the easiest.
Well, my lifelong dream is the genotype restaurant where your food is tailored to your taste genome.
Yeah. Interesting. Very, very, you know, I was aware that these differences exist, but they are so profound. It's absolutely amazing.
Aruba, did you have a question?
I did. So Danny, that was a very interesting talk. And but you know what I was really thinking is that I was looking at those funnel plots, and based on the sort of shape of those funnels, how many people really, I mean, if it were not for the extremes, would you capture any difference? I mean, I think what I'm trying to get a sense of is that how big a difference does it have to be? I mean, if someone has a problem with the bitterness gene mutation and they cannot taste anything that is bitter, that I understand. But can someone be oversensitive to bitterness and everything tastes bitter to them?
Sure. So you can imagine for the 25 bitter receptors, they have many polymorphisms. They're very, very snippy. You can imagine that there are many people that drew a bad hand and have the most sensitive allele for all those bitter receptors. So that has got to be a tough experience when having so many things, particularly plant foods and medicines, are just excoriatingly bitter. You don't get a break. And there are many people like that. You may be familiar with the term super taster.
And I also want to ask you that you said that it's really the loss of smell that leads to the loss of taste, which happened in the case of the COVID individuals. And you did make a comment, and someone walked into my office just then, and I missed that part. How is it reversible? Why does it become reversible?
So this is a big topic. So people lose their sense of smell and their sense of taste, and it is very reversible. And about 85% of people that do lose their sense of taste and smell regain it, and they get back to normal or near normal, but 15% do not. And what we think is happening is that there's something called a sterile infection. So the virus is cleared, right? There's no virus in the nose, there's no virus on the tongue, virus is gone. But the body is still engaging in those immune responses, and those immune responses prevent the taste receptor and the olfactory receptor cells from repopulating. And so we call it, the way I view it, it's a bit like a war where the enemy's defeated, but the other side is still fighting. And that's, we think that's why, and how to reverse it is not known. And I want to emphasize that because there is a lot of, I'm trying to be polite here, misinformation on the internet about regaining your sense of taste and smell. And there is very, very little the only thing that's known to help a little bit is the so-called smell training. And that helps some people a little bit, but there are no effective treatments, universal effective treatments. I'm sorry.
But this happens in the context of people who are getting chemotherapy also, or radiation therapy also. And for some of them, I mean, I know from my personal experience from my brother, he regained it. However, it seems to me that over many years, sometimes it does come back a little bit. I mean, these loss becomes permanent. And so it would be very important if we know how to reverse that. If it was there and it was lost, how to regain that. That would be a very important component of this one. So
yes, I completely agree with you, and we certainly are putting all hands on deck to try to reverse it.
So I have a follow-up question about that, which is, it makes me wonder about epigenetic effects or also gene regulation, because if I remember correctly, doesn't the taste perception change as you go from infant, child, adolescent to adult?
Yes,
right?
Correct. So genotype doesn't change, obviously.
Right.
So that's important to just emphasize. But what we do see is that children have more taste receptor cells than adults do. So it's partly that's an explanation. Doesn't mean that epigenetics is not important and gene transcription is not important, but there's a frank loss of cells, as there is with vision and hearing.
And then Arupa mentioned people who everything tastes bitter, but as you know, we've done bitter taste perception tests in different ethnic groups in Africa. And I do remember a handful of people who were bitter blind, I call them, because they didn't taste anything. Like nothing tasted bitter for any of the different substances that we tried. But what never, ever occurred to me at that time, this was over 10 years ago, was the possibility that I now wonder if it could have been viral, induced due to viral infection. Right? Something that was like COVID-19. We don't even know about it. That's like, yeah, that's something I'd love to follow up on at some point.
Yes, absolutely. Go ahead.
Oh, I have a question. I was wondering, maybe you haven't studied that, but I was wondering that does diet actually change your taste perception? So for, ethnic groups that eat a lot of hot stuff like hot peppers and things, would that eventually change your taste perception with time?
So there's very mixed data on this. So what we know is that reducing salt in your diet makes you prefer less salt. So that's a very robust finding. But we find that changing the amount of hot in people's food doesn't really seem to push their gene transcription around very much. So I think it's a bit of a mixed bag about that. And with bitter, I think that would be a fascinating study that's yet to be done, which is to really expose people very chronically to like bitter plant foods and see if that changes their thresholds and their detection and their liking and their hedonics. I think that's a great study.
And are you, oh, I'm sorry, go ahead, Dorothy.
Oh, okay. Another question about your study with the 4 different drugs. Was there a baseline assessment to determine if there was any underlying issue as to the taste perception? Since it was like a Zoom kind of like, virtual thing, were you able to get a baseline assessment of the receptors in their mouth before they did the test, so it doesn't bias the result one way or the other.
Yes, so we have a few control solutions that we know are very robust, so sweet and salt. So we have them taste those and give us answers on that, to just be sure that they have an intact sense of taste, generally speaking. Yeah, that's a very standard kind of thing that we do.
And I was going to ask, I assume, or are you doing a GWAS? I don't know if the sample size is that huge, but given the range that you saw, that it has to be there has to be many genes, I would think, right? Or at least
yes,
several.
absolutely. So, we have about 400 and plus people, and so we are underpowered for a GWAS, but you never know.
It's not bad, because it may not be that
Mm-hmm.
complex, right? So you actually may have enough power to at least see some candidates.
Yes. I'm kind of like a rat pressing the button to see when the genotyping results are done.
Yeah, that's going to be really interesting, because it's also making me think about, there is so much traditional medicine. At least I work in different African countries, and people use different plants and herbs and barks and things like that. And it would be really fascinating to also see how people perceive those in terms of taste.
Yes, absolutely. It's really one of time when I was first getting into this area, I actually just went out into my yard and I started just tasting things and seeing what they tasted like, which was not necessarily a smart thing to do. But
when you say you just went to your yard, you mean you just started picking the grass, like some leaves?
And leaves and stuff.
I'd be terrified. I'd get the wrong thing and keel over.
I spit it all out. But yes, so much of the plant world is bitter. It's really sort of surprising.
Is anything known about people when they are sick, like influenza, or all of these things that we are talking about now, the infections, the viral infections? Is there anything known that there is any taste variation during the duration of those infections?
So before COVID, taste loss, taste on the tongue taste loss was very, very rare. So not the flu, not RSV, not other infections. So COVID is really a different thing. And we're really trying hard to understand what that thing is. Because it is, when I say it's rare, it's just vanishingly small. A smell loss with other viruses like the flu is not uncommon. You'll often hear people that have lost their sense of smell with a viral infection, but not taste.
Mm-hmm. Okay.
Yeah, a quick one again. Thank you very much, Danny. I'm just curious how much, the regeneration capacity of the specialized receptors, for example, is very common in Africa, or where place where you have high prevalence of HIV, for people to have any fungal infection, be it candida or whatever, which may have an impact on the receptors. And I'm just curious how quickly they will regenerate, and if you have had a control on those previous infections directly affecting the tongue. So that's a quick one. And the other one is when using drugs like TAF, I'm just curious, procedurally speaking, how curious and how much are we taking care not to provoke resistance by giving a small dose of a drug to a person who may have had a latent kind of infection. And of course, thanks for sharing the PCA. And I noticed that Liberia is a kind of outlier from the other African sample. And any observation on that one? Thank you.
Okay, well, that's a lot to unpack. What I'll say is that I thank you for your thought about oral infections and taste receptor regeneration. So in a healthy person, the taste receptor cells regenerate in 12 to 15 days. So they're constantly turning over. And I think there's no knowledge about how oral infections might affect that. like the yeast infection that you mentioned. So that I just don't know of any data that address that. In terms of the resistance question, that's really sort of not my playground. I really don't know much about that at all. I really deal with the medications in a sensory testing capacity rather than a clinical medicine capacity. And whenever there's puzzling African data like those Liberian data, I always know that I could come over to the Tishkoff lab and get some comfort and advice. So that's what I plan to do.
Glad to do that. Are there any other questions? Otherwise, we are just about at 4 o'clock, so perfect timing.
Thank you.
Oh, Aruba, did you have a question? Or?
No, I just wanted to thank her.
Yeah, that was
And me too.
fascinating.
Thank you
Really
so
fascinating
much.
talk. And we'll see everybody. We will be sending out an announcement about next month. I think is going to be Hongzi Lee. We'll send out an announcement about his talk. All right, everybody, take care. And thanks so much, Danny, for a great talk.
Thank you.
Oh,
All
you're
right,
very
bye,
welcome.
everybody.
Bye-bye, everybody.